Okay, let's talk about Unit 2. If you notice, it has to do with methods, so it's a lot lab-related stuff. Okay? So, in Unit 2, you will actually hear some things that you probably already heard about. In passing, as I was talking in the lab, you may have heard some of these words, but this is my formal discussion on it. We talk about something called the 5 I's of microbiology. So what one letter do you think those 5 words begin with?
I.
F, no, just kidding. I, good. That was a sick joke. Anyway, they all start with I. And the first one is inoculation. Believe it or not, you already did it. When you took the sample off of your desktop, remember? And you put it on your plate, that's inoculation. When you coughed on the plate, that's inoculation. 2 days ago, you did the aseptic technique, right? You took a culture from a tube and you put it into your sterile tube. That is also inoculation. So inoculation is the act of transferring a pathogen or a microbe or bacteria from one source to another. So in the lab, it's going to be mainly from a culture tube that I will be giving you into a sterile media, either tube or plate or whatever, right? So that is inoculation. What you are transferring is called the inoculum. So the inoculum is is the actual sample that's on your loop, for example. So remember 2 days ago, you sterilized your loop, right? And you went in, I got a sample of bacteria from your culture tube. The sample that's on the loop is called the inoculum. The act of inoculating is inoculation. So it makes sense. Then you guys leave them on the side for me to do the second I. But normally, if you were the one working in the lab by yourself there, you would be doing the second I, which would be incubation. And hopefully you know what that is. You're putting them at the required temperature. You're giving them the proper type of gas that they need, the food that they need, the pH that they need, everything that they need, that's what you're providing them. So you're providing them an environment that helps them grow and multiply. So, standard, inoculation followed by incubation. And then you guys did it, isolation. Remember when you used the quadrant streak method? That was only 2 days ago, so hopefully you remember. You inoculated your plate, you did the Z, the tight Z, you turned your plate, did another one, turned the plate, did another one. You did 4 of them. And the purpose of that was to go from a mixed culture into a pure culture. You are isolating the bacteria from the mixed culture into single colonies. So that is called isolation. And we'll show you a couple of techniques here that are commonly used, but you used one of them. And that is the quadrant streak method. You already have done this actually, and you will do a lot more of it next Tuesday and the Tuesday after. Inspection. Didn't you guys look at your bacterial colonies? You did. And you saw, remember the head sample? You saw some white ones, some clear ones, some yellow ones. Just simply looking at them is inspection. Just simply looking at them. Now, you're going to look at them a lot closer when you use the microscope. You're going to be able to see their shapes. You're going to be able to see their arrangements. All those things about them. You're going to be able to see if they have a flagella or not. All those things fall under the word inspection. And then the final one, identification. Identification implies that you are looking at something or you're working with something that you don't know what it is yet. And you're going to do something, experimentation, to find out what it is. Find out what it is, ID it, identify it. Why is that important? Well, think about it. If a patient comes into a doctor's office and the doctor has no clue what this patient is sick from, what will the doctor prescribe? I don't know.
Nothing.
I don't know what I don't know what to give them if I don't know what it is. Example. If the doctor knows you're sick and they can't tell if it's a virus or a bacteria, they should not give you an antibiotic, because antibiotics do not work against viruses. They don't. But they work against bacteria. But on top of that, they also need to know which bacteria. Let's say they figure out, yeah, it is bacterial. Okay. Just give them this antibiotic. Random. No, they can't do that. Because not every antibiotic works against every bacteria. So they need to know even what bacteria you're suffering from, so that they can prescribe the right medication. So the crucial part for identification is proper treatment. If a doctor guesses that you have bacteria and he or she gives you an antibiotic, but it's not bacteria, you're actually sick from a virus, you know what they just did? They just made your virus episode, your illness, last longer, because your immune system weakens every time you take an antibiotic. It becomes weaker. The immune system that you're relying on to fight the virus. But you're getting the wrong treatment. So hopefully, and nowadays, that's what they did. They're not eager to give you an antibiotic. Now, a lot of people go to the doctor with the expectation that they're going to walk out with something. You see that, man? And they're very disappointed when they hear it's just a virus. And it becomes more of a symptom control kind of thing. Oh, I paid $200 for him to say, it's just, he didn't even give me an antibiotic. Well, he shouldn't give you an antibiotic because antibiotics don't work against viruses. But again, a lot of us go to the doctor hoping that you walk out with something that you can take and hopefully feel better. In the case of viral infections, your body is the one that's going to be mainly picking up the fight here. And all you're going to be doing is basically trying to control symptoms. So these things are very important. The identification is very important, again, for the main purpose of proper treatment. All right, so what I'm going to do now is quickly go through each one of them and see what else I need to add under each category. All right, this is inoculation. It is a handful. But when you go through it, you'll realize, actually you did a lot of these things, but let's see how I can guide your attention to at least certain things. Remember these things? These are your tools. Inoculating loop, inoculating needle, the cotton swab, the Pasteur pipette. These already come sterile in their own package. So you already have used them, you're familiar with them. The media that will be coming at you, they can either come in liquid form, and that is broth, liquid form, and you've used some already, or in solid form. Now, with respect to solid form, You can either have a slant, like you've seen, you saw a slant too. Or you can have something like this, where it's not slanted. So how do they do this? Watch this. All three of them actually start as liquid. All three of them. The only difference, listen, nice question on exams. The only difference between this media and this media, it's the same food. It's the same media. The only difference is you added something to this one that this one does not have. And what is that? A solidifying agent called agar. AGAR. That's the solidifying agent. It's kind of like Jello. Again, I don't know how to make Jello, but I know how to eat it very well, right? But from my understanding, you heat it up and you wait until it cools down. As it cools down, it solidifies. Am I right? I think I am. Same idea. You add the solidifying agent in there. Actually, to dissolve the agent, you have to heat it up. Well, yeah, you're going to heat it up anyways to sterilize it. So no big deal. So during the sterilization process, we mentioned already, there's heat, there's moisture, and there is... Oh, you're waiting for me to say it. What's the third thing that comes at it? Heat, moisture.
To sterilize it.
Yeah, to sterilize something.
Oh.
When we put something in the autoclave is going to come at it in three ways: heat, moisture, and pop them like balloons. How do you do that? You add pressure. There you go. Heat, moisture, and pressure. So anyways, during the sterilization process, you're already heating it. So the solidifying agent called agar will melt, will dissolve. So all the stuff is going to come out of the autoclave liquid. Then the solidifying agent will have dissolved in there. But if you leave it alone now, if you leave the tubes alone, the liquid one that does not have agar will stay liquid. The one that has agar will solidify. As it cools down, it will solidify. Okay, so how do I get this versus this? It's so easy. While it's still hot, you keep this tube like this. Just leave it alone. And then when it solidifies, it will look like that. All right, what about this guy? When it's hot, that means it's still liquid, right? You put it on a rack that has it like this, at a 45-degree angle. That's simple, it's nothing, no big deal. And it will, of course, form that media where it's kind of slanted, and it will solidify like that. So they have special racks that allows them to put the tube at a 45-degree angle or so to create that slant. But you gotta know that initially, all the tubes are liquid. If they have agar in them, they will solidify. If they do not, they will stay liquid.
Professor Saad, hold on a second, I'm just lost. So you say inoculation, the media can come in liquid form, the broth, solid form, stab tube, and stent tube. And you say they are the same, the only difference, you just add the solidified agar?
Yeah, the solidifying agent was the agar. So this has agar and this has agar. The only difference between these two is one of them you left it like this for a certain purpose. And the other one you decided to leave it like this for a different purpose.
So they call this a slant?
Yeah, the slant because you slanted them to solidify. Yeah.
Okay. Thank you.
So we finished this one right there. There are some media where they're in between the two. You look at it, you're like, that's not a liquid. Well, that's not a solid either. So can you guess which one that is? It's not a liquid, but it's not a solid. It's halfway. Semi-solid. And there's a purpose for that one. So how do I do that? Very simple. Very simple. If I remember right from my research years, we used to add Oh man, this is testing my memory. We used to add about 15 grams of agar per liter of media. I'm gonna double check and see if my memory is still there. 15 grams per liter. It will give you this. So if I want it not solid all the way, but not liquid all the way, what do I do? There it is. Add less. Add 7.5 grams per liter. Do you agree? It will solidify, but not all the way. But it will also not be liquid all the way. And we call that semi-solid. So, what is the purpose of that? What is the purpose of that? Where is mine? Look up here, please. When you have a, how do I turn this on? Sorry, there we go. Now it's following me. When you have a semi-solid tube, again, it's not solid all the way and it's not liquid all the way. That's when we use this guy, the needle. We're going to use the needle for a certain purpose. We're going to use it to stab this media. So using the needle, you're going to go in there and stab the media like this. Straight down and straight back out, you see? Of course, you'll have an inoculum on your needle when you do that. So a day or so later, look at this. Where you stabbed it is where you're going to get the bacteria to grow. Do you agree? Good. If it has no means of going anywhere, if it cannot go anywhere, it will stay where you stabbed it. Am I right? But if it can go places, here's what it will look like. Again, it will still grow where you stabbed it, of course. But if it can go places, it will go there. And it will fill your entire tube. And it will become cloudy. So this one is not going anywhere. It has no way of going anywhere, where this one can swim. So what does this bacteria have that this bacteria does not have? What is it? Motility. But what structure?
Binary fission?
What is it?
Binary fission?
No. Structure. Say it. Flagella.
Oh,
Yeah,
flagella.
Flagella.
Movement, yeah.
I was hoping I don't hear cilia, and I did not hear so good. No cilia in bacteria. Did I make myself clear?
Mm-hmm.
So if this guy can swim, it has a flagella. If I can walk, that means I have legs. You got it? That's the idea. So this is a nice experiment to characterize your bacteria with what? Does it have a flagella or not? So you inoculate, you stab it, incubate, take a look at the tube the next day. If it looks like this, you say, no flagella. Done. But if it looks like this, yes flagella. Done. And you'll be doing that. Now, there is another way of confirming whether your bacteria of interest has a flagella or not, and that would be staining. You can put it on the slide and stain it, and we'll talk about that. Okay, let's go over here. The type of media. There are a lot of different kinds of media, a lot of them. But we categorize them. We broadly characterize them under two categories. One is chemically defined synthetic media. So what is that? Chemically defined synthetic media. That is basically the company making the media and telling you exactly what's in there. Get it? Chemically defined. They're not hiding anything from you. 2.2% sodium chloride, 1.2% glucose, whatever. So you read the label, you will know exactly what's in there. And that's what's called chemically defined. And it's synthetic, so it's synthetic media.
Do you say the company?
Well, you're getting these from companies, regardless. The difference is that one company, for example, can make a set of media that they tell you exactly what's in there. And that's called chemically defined. The same company, actually, can make another type of media, and they will write like this on the bottle. Watch this. Glucose. Yeah, it's in there. How much? I don't know. It's in there. Protein? Yeah, it's in there. How much? I don't know, but it's in there. So you can tell that it's not, they're not really sure what's in there, but they know it's in there. How much is in there, they don't know what's in it, but they know it's in there. So that is called complex undefined media. And there's use for this one and use for this one. I'll tell you something. The 8 plates that you got on the first day of lab were this kind. Complex undefined media. What is the analogy between the two? Listen to this one. Hopefully it helps you understand. You are throwing a party. And you want the people that you invite to have something to eat. So let's say you invite 10 people. But before they arrive, you ask each one of them, what do you want to eat? One person says, for example, pizza. Okay, pizza. Pepperoni or sausage? Pepperoni. Okay. What do you want to eat? Fettuccine Alfredo. Okay, fettuccine. And so that would be this. That would be providing your guests with exactly what they want. Because the guy who's going to eat pizza will not eat the fettuccine, right? And vice versa. Because this one doesn't like that one doesn't. So you have to eat what you want to eat to survive. That would be this one. Compared to you throwing a party, and you really don't know who's coming. You know there are people coming, but you don't know who's coming. You're inviting the whole neighborhood. Hey, party, yeah, come, whatever. I got all the food you can ever ask for. That's this one. You catching the analogy? Mm-hmm. You have no idea who's coming to your party, and you want to make sure they have fun. So what are you going to have? You're going to have a bunch of pizza, fettuccine, hot dog, caviar, everything, lobster, everything. There it is. Eat what you want. That's this one. So I ask you this question. Not that last Tuesday, the Tuesday before, when you guys picked bacteria from the countertop, when you scratched your head, when you touched the plate, when you coughed, did you know who was coming to the party? No. Did you know what you were inoculating on that plate?
No.
You did not.
Undefined.
You had no idea. And that's why you use this one. You catching it now? Mm-hmm. When you want to just grow bacteria, you don't care what you're going to grow. That's what you use. Because you have no idea what's on your desktop. But if you knew who was coming, you would use the chemically defined to make sure they're happy.
To specify.
Are we good with the analogy?
Yeah.
Good. There's a question in your lab manual about this. And you'll hopefully understand what I just said and answer that question properly. Why did we use what's called general purpose media? See, watch this now.
To grow very
This is the same. When you use general purpose media, you're going to use this. You're going to use undefined media.
A variety of bacteria?
Yeah, to grow a variety of bacteria. Because you don't know who's coming to the party and you don't know what they like to eat.
Yeah.
So you give them all the food they eat.
So that's also known as general purpose.
Yeah, so now what we're going to do, keep your head up please. What we're going to do is reclassify the media now. These are the two main categories. But now we're going to categorize the media based on their function now. So it's like looking at these, okay, how do we use them? That's this one. Or what do we use them for? That would be this one. Well, in some cases, we use media for general purpose. That's their function. I'm just growing them. I'm just growing bacteria. I'm not doing anything special with them. That's this one. And this one will be the complex media. If you're just growing them and you don't know what you're growing, well, you better use the complex media. You agree?
Yeah.
So a lot of the things that we do in lab, that's what we're doing. General purpose, complex media. Now, if you know Dr. Saad is coming to your party, you better provide him something that he likes. I mean, there's not a food out there that I don't like. So that you're okay. But what is it that Dr. Saad likes a lot that I better have so he can have fun?
Heineken?
Yeah. He knows me very well. Heineken. There you go. So you're going to have a whole table of Heineken just for me. Oh, I'm going to be very happy. What did you just do to me? You enriched my life with Heineken. So in some cases, bacteria, they say, you know what? I can grow on this media, yeah, but I'll grow better if you enrich the media. If you add something in there that helps me grow better. That in the world of bacteria, it's not Heineken. In the world of bacteria, it could be, where's the enrich here? It could be growth factors, different amino acids, and so on. So some bacteria say, I will grow better if you give me, how much, 1% amino acid, 1%. No, I need 3%. You see? Your average bacteria can grow on 1%, not me. I need 3%, for example. Oh, the average bacteria doesn't need this growth factor, but I do. Do you see what I mean? So in some cases, the bacteria requires you to add something into the media to enrich the media for them to grow better. So if you invite me again to the party and you say, I am not coming unless you have Heineken, you're going to call me back and you're going to say this to me. You are so finicky. And I say, what'd you call me? Fastidious. Fastidious bacteria. Picky eaters. You can say that. Require enriched media. Okay. Here comes the fun part. Selective media versus differential media. You're coming into the part of the lab really soon where the fun really begins. So far, we're taking it easy on you. I think it starts not this Tuesday, the Tuesday after. The Tuesday after, we're going to take out the selective differential media plates and have you inoculate them. And that's when the fun really begins in the micro lab. So what do I say to my students? I say to my students, micro lab, is like a roller coaster ride. It starts really slow. And you're like, it's not so bad. Why is everybody worried about this ride? And it goes up, okay, hang on, where are we going? And it just takes off. It's gonna take off once you see these two come out. It's gonna take off. Enjoy the ride. Make sure your seatbelt is on. And how do you enjoy the ride? You come to lab prepared. That's what you need to do. You're gonna have 6 different selective differential media plates. You need to know everything about them. And we'll talk about it. You need to know what they do, what they kill, what do they not kill, why, and all those things. What color will be produced and why that color, and what color will the other one have and why that color, everything about them. And we'll talk about it when the time comes. But I'm just warning you. It may seem like microlabial is not that bad in the beginning. But when these start coming out, that's like an alarm. Uh-oh, things are about to get interesting. So come prepared. So this one, I think most students will understand. Selective media. It is the same, I can actually use the same exact media, this one. Same exact one. To make sure the bacteria has all the food that it wants, right? But then add something to it. And what would that be? An antibiotic? So that's what the lab staff will do. They will take the same exact plate, kind of like what you used on the first day, but before it solidifies, they actually on purpose add some kind of antibiotic in there. Or some other thing in there, a selective agent. So they're gonna on purpose add a selective agent, something like this, phenylethyl alcohol. The alcohol or the salt, mannitol, or the antibiotic, those are all referred to as, the alcohol, the salt, and the antibiotic are all going to be referred to as selective agents. They kill. They kill who? The sensitive or the susceptible bacteria. If the bacteria on your plate cannot handle the salt, it will die. If it cannot handle the alcohol, it will die. If it cannot handle the antibiotic, it will also die. So what did you just do? You made a selective media by adding into it a selective agent. And the selective agents come in varieties of things. And it's gonna start selecting who's susceptible and who is resistant. And students get these two words wrong. Susceptible comes from the word kind of like sensitive. I can't survive that. So susceptible dies. Resistant, I can handle that, lives. So a resistant bacteria will not be hurt by the antibiotic. A resistant bacteria will not be hurt by the salt or the alcohol. Well, that depends on which bacteria we're talking about. So some bacteria are resistant to alcohol, but not the salt. Some bacteria might be resistant to all the three examples I gave you. Some of them might be susceptible to all the three examples I gave you. We're going to find out when we set up those plates. Do we understand this one?
Mm-hmm.
The plate is going to select. It's kind of like, for example, let's say, for example, 5 of you do not show up to class with closed-toe shoes, to lab. I can now start selecting between you. Who has closed-toe shoes, you get to stay in the lab. Who doesn't, I remove you. So who am I? When I start doing that, who am I? I am the selective agent. What am I doing? Selecting. Based on what? Who has closed-toe shoes and who doesn't. You agree? You see that?
Mm-hmm.
Very good analogy. I just thought of it.
So that's kind of like how, in a way, when the lab comes back for kind of like how to choose antibiotics. Like, okay, so this is what
Of course.
this is what they were susceptible to, this is what they were resistant to, and then the doctor says, okay, this is what I'm going to prescribe.
If they know what you're sick with, they kind of know what they.
Yeah.
But if they identify a new strain or something going on with you that is not responding to the expected
Like when
to
they
the
send you
normal
into
antibiotic,
culture.
yeah, then they have to isolate the pathogen, plate it on the plate, and figure out what they use. But they wouldn't do it this way. There's another procedure for that, and you guys will be doing that one too. All right, it's called the Kirby-Bauer method. We'll talk about it.
Okay.
This is the one students get kind of like, listen, so you don't fall into that. Do you agree under selective media, somebody's dying?
Mm-hmm.
Yeah.
I agree. Here, no, nobody's dying under differential media. Nobody's dying, because that's not the purpose. So what is the purpose? The purpose is to, let's say we're all a bunch of bacterial colonies on the plate. The purpose would be to find out who among you is fermenting a particular sugar. For example, like mannitol, for example, that's a sugar. So out of all the bacterial colonies on the plate, yeah, who's fermenting the sugar and who is not? So what are you doing? You're distinguishing, right, between the colonies that are fermenting and the colonies that are not. Another word for differential is to distinguish. Remember it, because it's going to come back at you in multiple ways. You're going to replace the word differential with the word to distinguish, because we all know what distinguishing means, I hope. Differential is kind of like, I don't know what that is. It's the same thing. To differentiate between, I can do it based on gender, I can do it based on height, I can do it based on whether you're fermenting or not, right? Is to distinguish. And that's what you're doing on this plate. You're trying to figure out who's doing what and who's not doing it. So obviously, in the world of bacteria, that's a biochemical event. Some kind of biochemistry is going on in this colony, but not in this colony. You see that? And during that week when we're doing this, it's going to be mostly about fermentation of different sugars. So we actually are going to set up this plate. Oh, wait a minute. You're going to set up this plate, but you just said this guy is selective, the salt. Am I right? But you're just telling me that this guy is differential. Didn't I just say that? Mm-hmm. The sugar is going to be used by the bacteria, either fermenting or not. That's differential. But the salt is going to be doing killing. I'll get to your question. The salt is going to be doing killing, right? Oh, so wait a minute. So the plate that has salt and mannitol is both selective and differential. You catching it? Mm-hmm. If I only put salt agar, that's only selective. If I only put mannitol agar, that's only differential. Mm-hmm. But you're going to do this one. You're going to put salt and mannitol. You're going to put a selective agent, the salt, and a differential agent, the mannitol. So some of them are going to die. Who's going to kill them? Let me do it again.
The salt.
On the salt mannitol agar, you're putting both the salt and the mannitol. Mm-hmm. Some are going to die from the salt. Yeah. Out of the ones that survive, you will check and see who's fermenting and who's not. Are we catching that?
Who's fermenting the sugar?
Yeah. So I'll give you an example. When I go look at your shoes on Tuesday, if I find you wearing open-toe shoes, I am removing you from lab. That's equivalent to the salt doing its job. Am I right? You are no longer in the lab. So let's say I have 15 students left in the lab. Now what will the differential agent do? It will look to see who's working and who's not. You catching it? Mm-hmm. You're in the lab, but some of you might be just sitting on a chair doing nothing. That's equivalent to non-fermenting. And some of you may be actually working. That's equivalent to fermenting. We catching the analogy, I hope? Yeah, go ahead with your question.
Okay, I was just lost. When you use differential media, it's only to see
a biochemical event.
Okay.
In our experiment in the lab, is going to be mainly fermentation.
Okay.
Mainly fermentation.
So you say something for buffalo then when you have salt and mannitol?
Then you will label the plate selective differential. It's both.
Oh yeah, you will distinguish who does this, who does not.
Yeah.
Who
The
killed,
salt
who
is
not
going to
killed.
kill,
Okay.
and the mannitol will be fermented or not.
Okay.
Yeah. Out of the 6 plates that you will prepare, you will only have one of them that's like this, selective only. Only one of them will be selective only, and it's actually this one right here. It only has the selective agent. That's it. The other 5 are going to be selective and differential. And this is one of them, one of the ones you're going to use.
So when you add the salt, like even though it kills, like is it possible for, like small traces of it to be left?
Traces of the bacteria?
Yeah. Like,
Now, that's a good question. Now, if we add the salt at the concentrations that is expected, no. You will see who survives the salt. I'm not going to give everything away. During the lab, I'm actually going to expect you to come with that information even before you touch the plate. You'll see, it's called the pre-lab. That's the whole idea of a pre-lab. You have to answer most of the questions about all of this even before you touch the plate to analyze it. Why? Because if you don't, you're going to look at the plate and you're going to do this. What? I don't know what's going on. I don't know what selective is. I don't know what differential is. What is salt? What is mannitol? I have no clue. And we only have limited time for you to figure it out, and I'm not going to wait. When the plates come out for you to analyze, you're probably going to have like 15, 20 minutes. That's it, we're done. I'm not going to sit there waiting for you to figure out what's going on. We don't have time for that. The figuring out what's going on, that happens at home. We call that the pre-lab. Do we understand that? I told you this is not Gen Bio anymore. You got to come into the lab fully prepared, knowing exactly what you're about to do. You actually will need to know even or predict the results. You'll see. And then you come to lab and you say, yep, I knew that would happen, and you're done. You're just confirming what you come in knowing was going to happen. Completely different than trying to figure things out on the day of the lab. It's not going to work for you. Do we understand the difference between selective and differential? Any other questions? Okay. The rest of these are very straightforward. I know you can read them easily, but let me just go through them real quick. Anaerobic, media that is free of oxygen. In some cases, you do have to grow bacteria that are anaerobic. That means you have to remove the oxygen from the media. So this is also referred to as reducing media. Reducing media. We will use one of them. Carbohydrate fermentation. Again, we will use those two. The media has one type of sugar in there. For example, glucose. In another tube, sucrose. In another tube, whatever, mannitol. And you're checking to see, does your bacteria like sucrose? Glucose? Mannitol? Or is it like Dr. Saad, eats everything? Right? We'll see. We'll find out. So that's called carbohydrate fermentation. Can it ferment this one, or this one, or this one, or all of them? There are some bacteria that can ferment an array of sugars. But there are some that are very specific to which sugars they can ferment. We'll be doing that too. If you want to send your bacteria across to another country, for example, you're going to take a sample of it, put it in what's called specimen transport media, just to simply send it to another lab, for example. The assay media, that's what you were talking about when you were talking about checking to see if a bacteria is antibiotic resistant against one antibiotic or another. That's called the assay media. We'll be doing that too. The enumeration media, this is designed to count bacteria in a given specimen. They typically use this type of media when we're talking about things like milk and beer and stuff. They cannot sell you these things if they have, if the number of bacteria in the milk, for example, is higher than what is allowed. So they have to do this to confirm that they are staying within the expected regulations. Once they do that, they figure out, okay, we're good, they bottle it and they send it to sell to you. All right, questions? Okay, let's see what else. Incubation, very straightforward. Incubation, basically, you need to provide the bacteria the proper temperature, the proper gas. A lot of times it's going to be the oxygen, but sometimes actually bacteria want CO2 as well, or a mixture of the two, things like that. So it depends on what the bacteria is. If it's aerobic, then it needs oxygen. If it's anaerobic, then it cannot handle oxygen. Oxygen can actually possibly kill it. And the amount of time, the timing. Sometimes you can grow something for a day or two, sometimes you may need longer. So this is collectively we refer to as incubation. Isolation. You already did this. Well, you have it set up, but we'll look at your plates. Remember this? The quadrant streak plate method. I'm not talking about it because you actually did it.
Mm-hmm.
But the main purpose of it again is isolation. To go from a mixed culture to a pure culture. Does anyone remember the other word for pure culture?
Arsenic.
No, not arsenic. Arsenic will kill you. Try it.
Oh, no, it's not arsenic.
I know. What's the word?
Arsenic.
Axenic.
Yeah.
I know. Arsenic you want to stay away from. All right. Axenic. Very good. But then there's another method. It's called the pour plate method. Why? Because you're literally pouring it. But listen to this one. You make the media. It's still liquid, but it will solidify. But it's still liquid, and then you inoculate it with a certain amount of your culture. And then you dilute it out by taking a small amount of this and putting it here. And then you dilute it out. And then you dilute it out. Now you have 4 tubes. Then you pour each one of them. You see? So they're called dilutions or serial dilutions. And then you grow the plate. And you'll get this. Obviously, the most concentrated one will produce the largest number of colonies, and the least concentrated one will produce the lowest number of colonies. There is a purpose for this. I mean, I tell you, I would not use this technique for your standard isolation. Oh, that's why we didn't use this, because we're just isolating. That's why we use this one. This one is more commonly used. Watch this. When you are growing or counting microaerophiles. Let's look at this word. A microaerophile is a bacteria that can handle oxygen. Watch, in small amounts. Micro, I like oxygen, aerophile. But don't give me atmospheric oxygen. The oxygen that you're breathing in this room is 21% oxygen. It would kill them. It's too much. So if we do this plate, if we did this plate, I wish we had actually, you would notice something. There'll be no bacteria growing on the top of the media. You see that? Because what's on the top of the media? Oxygen from the air. So if you look at the plate, the oxygen in the air is going to hit the surface of the plate, kill everything. So what's the, but you have colonies here. They're not on the surface of the plate. They're actually within the media. They're inside the media, where the concentration of the oxygen is lower. You catching it? Oxygen, look over here. Oxygen cannot penetrate through the media. It can hit the surface of the media, kill all of them. But it's the deeper ones. Remember, you're mixing the bacteria. You're gonna mix it. So you're gonna have some that end up on the surface, but a lot of them are gonna be embedded within the media. It's those ones that are gonna grow. The surface bacteria will not grow. So this is used when mainly when you're using microaerophiles, bacteria that like oxygen, but in small amounts, in small concentrations. 21% oxygen, which is atmospheric oxygen, is too high for them. It kills them. All right, here's another approach, spread plate technique. I've used this a lot in research, but the drawback to this, you have to already know what dilution that will give you a good isolation. You see? So you have to already have some knowledge on what dilution do I need to add to this plate that will give me nice isolation. But how do I add the sample to the plate? It's very easy actually. You put the sample in the middle, watch, and you literally put it on what's called a turntable. It's a little round table that turns around, that spins. And I used to have fun with this thing. You put it on there, and you got an L-shaped glass rod that is sterile, and you put the glass rod to the top of the plate, the media. You touch it, and then you spin it like that, a couple of times. It spreads it out. Very easy to do. But the drawback to it again is you need to know what dilution to use to get good isolation. So if you just standard, I just want to isolate, it's this one. You see? And that's why you guys use that one. Inspection. Maybe finish the 5 eyes and I'll give you guys a break. Inspection is looking at things. And you guys will be doing this stuff soon. Actually, this is on Tuesday. You remember your broth tubes? You inoculated them with the aseptic technique. Some of them may grow like this. They only grow on top. They're actually on the top of the medium. They're not swimming like this one is. We call this one turbid. We call this one pellicle. So what are we calling? We're not calling the bacteria that, we're calling the type of growth. In broth media, liquid media, you can have different kinds of growth. One of them, where it sits on the top, that is referred to as pellicle. The other one, where it goes all the way through, that is called turbid or cloudy. Now, why does this one only sit on top and it cannot mix with the media? Because think about it. The media is mostly water. And there's something in the cell wall of this bacteria that is hydrophobic. So the bacteria has a hydrophobic substance in its cell wall. And because of that, it cannot mix with the water. You catching it? It's like oil at this point. Does oil mix with water? No, because it's hydrophobic. So, for example, if this is hydrophobic, and I have it attached to my wall like this, and I have like hundreds of them, you throw me in water, I'm not gonna go swimming. Why? Not because I don't want to go swimming, no, it's because I can't. This is hydrophobic. What is this? It's called mycolic acid. You're gonna run into this real soon. Mycolic acid. So bacteria that have mycolic acid, like the mycobacterium, will grow like this in liquid broth. The mycobacterium that causes tuberculosis, the mycobacterium that causes leprosy, would grow like this in liquid broth. Because why? Because they have mycolic acid, making their cell wall hydrophobic to the point where they cannot mix in the liquid of the media, which is water. Okay, how about the ones that can mix? Well, obviously these three can mix. Well, this one, the difference between this one is this one can swim. So it has what? If it can swim, it has what? Flagella. Yeah.
The fact that they are hydrophobic and they are related to like leprosy and you said, was it tuberculosis?
Tuberculosis are not related. The mycobacterium that causes leprosy and tuberculosis will have the mycolic acid.
Correct, and thus they are hydrophobic.
Yeah.
The fact that they are hydrophobic, is that a correlation to how it affects your health? Like if a bacteria, when it has the mycolic acid and it's microphobic, it's a direct correlation to
Mhm.
how it affects.
It could be, but like tuberculosis, for example,
Mhm.
it likes to isolate itself in your lungs.
Correct.
So it's gonna build a little chamber where it lives in. So it doesn't even see what's going on outside. And then from time to time, it causes problems.
Yeah.
It could be.
So does it mean that because the turbid one can swim, it's hydrophilic?
Oh, yeah.
Okay.
Yeah, you can't mix it in this water unless you're hydrophilic,
Okay,
yeah.
okay.
Which means it doesn't have mycolic acid.
Okay.
Yeah.
Yeah.
So if you have mycolic acid, you're gonna do this. If you don't, you will be one of these three.
Mhm.
But now what's the difference between these three? This one has a flagella. It can actually swim to the point where it makes the media cloudy. Can you tell me what this one does not have?
Flagella.
A
It
flagella.
doesn't have a flagella, yeah.
It feels the force of gravity and it falls to the bottom and it says, oh, too bad I can't go anywhere. So you're gonna see them settle to the bottom. You actually used one of those last week. And you have these guys, they produce clumps. They're sticky. They stick to each other. Kind of like if we were 3 different bacterial cells of this variety, we would stick together as a group and we would go together as a group. So we'll talk about it, but they produce an enzyme that makes them sticky. And that's called clumpy or flocculent. So you need to know these words: pellicle, sediment, turbid, clumpy, flocculent.
So clumpy like has a flagella too, or it doesn't have?
Not necessarily, no. They produce an enzyme that makes them stick to each other. Yeah. So they're harder to go independently on their own way. They travel as a group together. Yeah.
And what was the reason that they settled to the bottom?
Oh, this one? They don't have a flagella.
Yeah.
Okay, so that's one way to do inspection. You'll be doing it on Tuesday. You're gonna look at your tube, you're gonna say turbid, flocculent, and so forth. But I will tell you something. They're gonna be mostly this or this. Why? Because we did not set up these two.
Oh, okay.
So on Tuesday, you'll either have this or this. We did not have examples of these two to set up on Tuesday. All right. On solid media, you're gonna now see the colonies. Or the actual, in your case, the slant. Am I right? Didn't you do the slant?
Yeah.
So when you did the slant, you inoculated the sample on the slant. But the way we did it, I'll tell you something. The way we did it, we did the zigzag. Remember that?
Mhm.
Because we did that, and I really don't care for you to know that detail, you will really won't be able to analyze these this way, but we'll talk about what they are. Typically, to do this, it's best to do this kind of thing. Watch, where you have the tube, it's a slant. You only inoculate like that.
Mhm.
You go down and you can just streak out
Mhm.
like that. Instead of zigzag out, you do a streak out like that. Then, 2 days later, you look at the margins. You look and see, are the margins smooth? Do they have smooth edges? Are the margins irregular? Do they have what looks like a filament or a rhizoid? We're not doing all of that, all right? For you to do this, I have to set up all the different tubes that give you all the different possibilities, and there'll be plenty of them. But the thing is this, your guys, most of you, they're not gonna be microbiologists. You're gonna be nurses, and you're gonna never have to do this. So we'll get an idea of it and know it's out there. If you do become a microbiologist in the future, you will be a professional at this. So what are we doing with this? We're characterizing this growth. We're gonna characterize it first based on opacity, meaning, can you see through it? If the answer is no, I can't see through it, then it is opaque. That one you can do. That one you'll be able to do on Tuesday. Can you see through it? You'll be like, I don't even see anything. Yeah, maybe because it's translucent or transparent. So there are in some cases, you'll come to me and say, Dr. Saad, I think they forgot to inoculate this. Because you get so used to the opaque ones, the ones that you can simply see. And I say, no, it's there, trust me. But you can't see it because it's translucent. So those are the two options when it comes to opacity. It's either you can see it or you can't see it. If you can see it, it's opaque. If you cannot see it, then it's either transparent or translucent, and the difference between these two is a very fine line. Margins, I just told you. Does it have a smooth edge? Here. Does it have a smooth edge? Does it have an irregular edge? And so forth. Texture. You will actually look at it and see, is it shiny? If it's shiny, it's moist. It has water in it. Or is it crusty? If it's crusty, it'll be dry. And this one you'll be able to see easily. You already saw it. Does it have color? Is it pigmented or not? Some of them will be red, some will be yellow, some will be white, and all these different colors. And then, microscopically, under the microscope, you'll be able to do further inspection. Is it round? And that we call that coccus. Is it a rod shape, bacillus? Or is it spiral, spirillum? Those are the shapes, the three most common shapes. But you'll also be able to assess their arrangement. Do not confuse shape with arrangement, because a lot of students do. Shape is this. Is it round? Yes, round, coccus. And then when you look and you see, oh, there's two of them right next to each other that are round, attached to each other. Oh yeah, because they're diplococcus. You catching that? The prefix is the arrangement. The suffix is the shape. Diplococcus, two round shaped bacteria. That's a diplococcus. All right, diplobacilli, two rod shaped. Strepto is a chain, kind of like a beaded necklace. Streptococcus or streptobacilli. Staphylo, kind of like grapes when you go shopping and you go to the grocery or the produce and you pick up grapes, not in the box, actually on the vine, right? It's a random arrangement of grapes. That's exactly what this is, Staphylo. But look at it, there's no Staphlobacilli. The Staphylo is for the coccus. You never see rod-shaped grapes. Think about that, right? Tetrad is 4 cocci together, like that. That's a tetrad, only cocci. Sarcina is 4 of them and another 4. Kind of looks like a Rubik's Cube, if you know what that is. It's 3-dimensional like that.
So what's the difference between
Between?
yeah, these two, like
This one and this one?
no, like the
This?
yeah
And
the,
this?
no, like the one
Tell me the name.
okay, Sarcina, yeah, this one, and the one stay
Staphylo?
yeah.
Oh, maybe we didn't draw enough circles, but this one is only like that, 4 and 4.
Okay.
This one is basically a bunch of grapes coming together, random and irregular arrangement. This has a regular arrangement. This would be at random, irregular arrangement. It may look like this, looks like this, but it doesn't. This is 4 of them lined up in front of another 4. We tried to make them as random as possible. Yeah. But you notice there's no bacilli here. And this one, there's no coccus here. This is like a picket fence, palisade. It is like this one, but folded over. Do you see that? So when it comes to bacilli, they can do this. They can take the bacillus and fold it over where you have what looks like a picket fence. When it comes to the spiral ones, you have the spirillum or the spirilla, and you have the spirochete. What's the difference between the two? One is not flexible, but the other one is flexible. So the spirilla will swim like this, corkscrew kind of thing, but it's very rigid. The spirochete will also swim like this, but it can bend. That's the difference between.
You say one is,
This one swims in a corkscrew manner, but does not flex or bend. The spirilla. The spirochete also swims in a corkscrew manner, but can bend or flex. And then you can microscopically look at any specialized structure that your bacteria that you're looking at may have. Things like the flagella, the capsule, and the endospore. Let me see what else we have, and I'll give you guys a quick break. Inspection of, obviously, the microscope. We talked about this. I'm not talking about this. Am I right or not?
yeah.
We're good?
Yeah.
Yeah. Let's stop here, give you guys a break, and we'll finish it.